cell walls, cell membranes, organelle membranes. They hold us together. The skin protects the insides from the outside. Intestinal linings absorb nutrition from our food while preventing it from contaminating other body tissue.
Membranes in the lungs absorb oxygen for use in respiration. Blood cells transport substances to cell tissues which form musculature, skeletal, and nervous systems, as well as other internal organs. At every step, cell and organelle membranes regulate the passage of substances throughout our bodies.
Did you ever wonder why cells in our bodies absorb some substances and not others?
This is a model of a simple cell or organelle membrane. In a simple membrane, substances can pass through if they are small enough to go through the natural openings in the membrane.
If they are too big, they won't go through. For example, water and carbon dioxide molecules are quite small and pass through easily, while glucose is too large and won't go through. Usually substances will move or diffuse from areas of higher concentration to areas of lower concentration. This is the principle behind osmosis as well.
However, a system like this is not very exclusive. Only substances that fit through the hole can enter the cell, whether they are desired or not.
In order to control the substances which enter the cell more effectively, nature has evolved a number of selective transport mechanisms.
There are basically two kinds of transport mechanisms. Ones which require energy to function, active transport, and ones which don't, passive transport.
Let's look at a couple of passive processes first. We'll add something to our cell membrane called a transmembrane protein. This protein is a complex molecule which extends through the membrane and acts like a keyed porthole into the cell.
The key in this case can only be provided by a molecule of the correct shape. This model is shape-specific to glucose. The glucose molecule will fit in here, but the sucrose molecules won't. Once a glucose molecule diffuses into this keyway, it causes the protein molecule to wobble or change shape. This causes the outer port to close and the inner port to open. The glucose molecule then diffuses out. The absence of the glucose causes the protein to wobble again, back into its original shape. The inner wall closes and the outer wall is ready to accept another glucose molecule. The second type of passive process involves a transmembrane protein called a voltage-sensitive gate or ion channel.
These are found primarily in the neurons and respond to various ion concentrations. Under normal conditions, there's a voltage differential between the extracellular fluid of the cell membrane and the intercellular fluid. Under this mildly charged condition, the voltage-sensitive gate is held closed.
The charge of the outer surface can be disrupted by electrical, chemical, or physical stimuli, depending on the purpose of the cell. When the voltage differential shifts, the voltage-sensitive gate opens and, in this case sodium, ions flow into the neuron. This causes the desired reaction to stimuli in the cell.
Now other transmembranes begin to pump out the high levels of sodium. When the normal concentration gradient for sodium is reached, the voltage differential across the membrane is restored and the voltage-sensitive gates close, ready for the next stimulus. In the last example, there was another group of transmembrane proteins which pump sodium out of the neuron and pump potassium back in. They're called sodium-potassium pumps.
This is a model of a sodium-potassium pump. It's a large transmembrane protein like those used in facilitative transport, except that in order to wobble or shift its form, it requires energy. Like our earlier example, it is also shape-specific. It has bonding sites on its interior for 3 sodium ions. However, the bonding of the sodium is not sufficient to cause the altering of the protein's molecular structure. The movement of ions against the concentration gradient requires additional energy. The energy comes from adenosine triphosphate.
It bonds an additional phosphate to the transmembrane in a process called phosphorylation. This produces the desired conformation. The transmembrane is now open to the exterior of the cell, and the sodium molecules are released.
The interesting thing is that not only has the change in shape of the protein inverted the gate, it has also provided bonding sites for 2 ions of potassium. The bonding of the 2 potassium ions causes the release of the phosphate and triggers the protein gate once again. The gate is open to the inside of the organelle, and the potassiums are released. The transmembrane is now ready to start over again.
It takes a couple of minutes to explain this process, but it isn't slow. Each channel in a cell is capable of undergoing this shift 100 times a second. Imagine that this cell contains a fair amount of glucose, and its purpose is to accumulate more glucose from the surrounding extracellular fluid.
The cell can't get glucose through simple diffusion. In fact, glucose wants to travel out of the cell along its gradient from high to low concentration. However, the facilitative transport protein and the sodium-potassium pump can work together to drag glucose molecules into the cell against its concentration gradient.
By pumping out sodium, the sodium-potassium pump creates a sodium gradient which favors the transport of sodium back into the cell. Sodium will diffuse back into the cell if given the chance.
Now, if we have a transmembrane protein which is shape-specific to both glucose and sodium ions, the sodium will diffuse in and literally drag the sugar with it.
The proton pump works in much the same way, except that it works on a proton or hydrogen ion gradient rather than sodium.
Proton pumps are found in the organelles of all living organisms and involve the use of 2 different transmembrane channels. Like all transmembrane proteins, proton pump channels are capable of conforming their shape in order to allow the passage of specific molecules.
The energy needed to accomplish this is provided by electrons and protons released through respiration in animals and through photosynthesis in plants. When protons are pumped out of the organelles, the result is a proton gradient which favors the movement of protons back into the organelle. If allowed to, protons will diffuse back into the organelle.
This process occurs in the second channel, called ATP synthase. The proton passes back through this transmembrane protein, and in so doing causes the production of adenosine triphosphate. If you remember, ATP was the substance required to power the sodium-potassium pump. So we've come full circle.
These 5 different transport processes occur in the living tissue of all plants and animals, including ourselves. As long as they work properly, we remain healthy and well. Some diseases such as cholera interfere with these processes.
The symptoms of cholera are diarrhea, loss of electrolytes, dehydration, and sometimes death. What is happening in terms of passive and/or active transport processes?